Welcome back. In this presentation, we will be talking about some of the considerations and factors that must be taken into account when we do analysis on nutritional data. Measuring and analyzing human diet is a complex task, and there are multiple factors that can affect the quality of the dietary assessment and could potentially influence the data analysis and the results. And even though we already discussed about the traditional assessment tools and its strengths and limitations, when we consider the structure, the content, and the presentation of the data collections of these instruments and how we decide to use one or another, we can end up influencing the quality of the data that we collected and could result in potential errors, missing, or skipped responses. And we need also to consider the relevancy and the validation of the dietary assessments used in particular populations, as the questions might not represent the usual intake or preferences of the population. So, as an example, let's pretend that we're going to use the EPIC-NERC-FOL study food frequency questionnaire in a cancer study in Laos. Even though the EPIC study is focused on cancer research, the target population are European individuals. If we use this FFQ of this particular study in our study, we will be missing information and questions that might not be relevant for Laos, as it responds to the diet and preference of European citizens, but probably will not capture Laos reality. There are other potential factors that we need to account before analyzing any nutritional factor. Think about the use of nutritional supplements, if there are specific characteristics on the individuals, such as allergens. There might be changes in the dietary patterns, and that can be accounted by multiple measurements, even by seasonality, or even the interaction with other substances and nutrients, such as pharmacological drugs or toxins. So all of the factors that I just mentioned can affect the assessment of diet, but it also accounts for the variability of our diet. So let's start with a quick exercise. Think about what you ate yesterday. How many meals did you make? What did you have in each meal? What were the quantities that you had for each of the food items? Now, compare yesterday's diet to what you had 2 days ago. 1 week ago? 1 month ago? How much did it vary? All right, now let's think about one of your friends. Did that person eat the exact same food that you ate yesterday? I'm pretty sure that there is a high probability that you're going to answer no. Let's get it a little bit deeper. Have you considered the thousands of substances that are in one food item? How those substances interact and can affect your health? But to make it a little bit more complex, there are no two food items with the same amount of nutrients. So when we think about the complexity of human diet and how it interacts with health, we must account for variation. And so with this little exercise that we just made, I just account for how diet varies throughout our life. There are two main concepts that we talked about with variability in diet. We can have everyday changes, and that's what we talked about in the first half, and that's called random variability. And it's kind of what you can see in the first graph. So every day, we change it a little bit, even though we have a usual intake. Now, there can be systematic variability, and this happens specifically with seasons, for example, or when you get sick. So think about like harvesting calendars. There are certain products that we eat more often when they are harvested than when they are not. This can account for the systematic variability. Now, as we talked about in the second part of the exercise, there is variation within, not just within person, but between persons. We don't need the same thing as our friends or as our family. There are always going to be differences. But now those variations will account at the end for the usual true intake of the population. And that's what you see in graph number 2. Also, let's consider the third part of the exercise that we just did. We were talking about the amount of substances that are in food and how much these substances and their quantity varies depending on the food item. And even though we have an item that has the same size and then the same weight as another, these nutrients are going to vary from one to the other. And this is due to other factors that must be accounted in diet variability. So among these factors, we have nature or true variation. And as the word says, this is how much a food item can vary depending on its ripeness, the time of storage and the place, the seasonality when it was harvested, and even the nutrients content in soil where it was harvested. Another factor that can affect nutrient variability in food is the processing that we expose to the food items. So let's think, for example, when we talked about rice. If we have brown rice, because of the minimal process that it's done, the amount of nutrients is higher than when we are looking at white rice. All the process of whitening reduces the amount of nutrients in rice. But there are also other factors such as measurement errors that we can see both in nutrient or content variability and also in diet variability. Some of the errors that we normally need to account for is for recall, when participants don't remember the nutrient or the food item that they are ingesting, the coding process for the assessments, and there are also human mistakes and lab errors that can affect diet and can create variability in our data. Now, why do we need to consider variation is because of two main reasons. First, because we are trying to assess the usual intake of populations, and second, because the associations of diet and health disease outcomes can be affected based on this variability. So for the first case, let's just remember the first two graphs that we saw in the previous slide. If we only account for 1 day of data or for only one intake, the chance is that we're going to have a poor estimate of the person's or the population's true long-term nutrient intake. But the estimate or the true intake can be improved by using the average of multiple days of data for that person or for that sample. Now, for true associations, depending on the type of variation that we're experiencing, then the result is going to change. When we have random variations, again, let's think of only having one measurement instead of multiple, the association between that nutrient or diet or pattern will probably be attenuated or closer to no results when we looked at the disease or the health outcome. For systematic variations, so let's think about seasonality, for example, then we can either see a direction or bias either to the upwards or downwards side. And so this means that when we look at the association, it might be exaggerated, it might be higher than what truly is, or on the contrary, it can be attenuated and can be lower than it should be.
And even though we have discussed in the past module and in this one that there are no perfect assessment tools for nutrition data, what it is important is that we need to understand what is the magnitude of error that we have for each specific assessment, but also how to evaluate its effect in the relationship to the research question that we are doing. Now, there are other sources of measurement errors, not just variability, such as misclassification of the individual's usual intake. And this could be an error on coding, could be an error on reporting, or it could be an error in the assessment. But also, we can talk about reporting errors where we have under or overestimated nutrient and food intakes from the participants. Now, the good news is that measurement error can be handled through statistical correction, but it is going to depend on the form of the error and how it affects the associations of interest. Now, let's talk about the second factor that it's fundamental to take into account when we're doing nutrition data analysis, and this is energy and nutrient intake. Particularly with energy, we need to have a special consideration in nutritional analysis or nutritional epidemiology, as the level of energy intake may be primarily a determinant on a disease. There are individual differences in total energy intake produced by the variation of the intake of specific nutrients that are unrelated to diet composition, but also energy intake may be associated with the risk of disease, but is not the direct cause. And with these three main points, let's just remember that energy intake is determined by body size, physical activity, and metabolic needs, which can also be related to the risk of the disease. On the other side, normally when we talked about total energy intake, there are foods and nutrients that are commonly associated with these. A higher total energy intake usually can be linked to a higher intake of other nutrients, particularly with macronutrients. But also, there are nutritional factors that can be analyzed in terms of absolute amounts or in relation to total caloric intake, which depends on the biologic relationships and public health implications. So, for example, if a nutrient of interest affects an organ not related to body size, like the central nervous system, then it would be better to measure the absolute intake of that nutrient. But on the other side, if the nutrient of interest affects a system that it's related to body size, let's say the metabolic system, then it would be better to measure it in relation to energy intake. And also, in some cases, energy intake might not be related to the disease. So in this case, the body size, the physical activity, and the metabolic needs are not determinants of that particular disease, but it contributes to the variation of the nutrient intake. So we call it extraneous. Now, nutrient intake is strongly correlated to energy intake, particularly with fat, protein, and carbohydrates. And by controlling the variation in total energy intake, we can control measurement error of these specific nutrients. So because of all of these facts, it is necessary that when we do nutritional data analysis, we adjust this analysis by energy intake. Now, just remember that it goes case by case, and in some cases, we need to account for the absolute nutrient intake instead of these relationships. Finally, let's talk a little bit about the issues of reproducibility and validity on some of the nutritional assessments. So before we talked about the different approaches and how it can affect our results, let's just consider that before the analysis, we need to make sure that the quality of the data is the right one. There might be missingness and blank questions. For example, when we have more than 100 item FFQ, we can have incomplete responses or with no detail, or even we can have data coding and entry errors. And even a small error such as changing a teaspoon for a cup might create a misclassification of that individual's nutrient intake. Now, the approach that we use to examine the health effects of foods must consider specific factors, and sometimes food groups or single food approaches might be inadequate because first, there can be specific food items that might be relating to more to eating pattern, and then we can end up having biased results. There can be biological interactions that we are not considered when we are looking at one single food item. So let's think about iron absorption, and vitamin C. There may be food groups or specific food items that may produce significant results just by chance. And then there can be high level of intercorrelation among some nutrients, like when we are studying magnesium and potassium. The good news is that there are methods that we can use to account for these issues. From the quality perspective, we need to make sure that our assessment tools are complete, are validated, and there are no missingness. And then we need to make sure and double check that data entry and coding is correct. From the statistical perspective, there are more comprehensive methods such as the dietary pattern analysis that can help us account for these issues. And also we can do a validation between different assessment tools such as the food frequency questionnaire versus biomarkers. Now, one of the things that I want you to remember is that when we're deciding upon the data analysis and even on the data collection, the first thing that we need to have, and this is something that we're going to study in a couple of presentations, is that the research question and variables must be considered before deciding on the approach. So basically, this means that before we decide on using a specific method or we decide on using a specific assessment tool, we need to know what are we asking for. And this is because sometimes when we don't have the correct assessment, the results that we get might be diluted. For example, think about having a folic acid and neural tube defect question. But then if you use a dietary pattern approach, your results might be diluted. All right, now it's your turn. I want you to look at the Laos Health Department dataset. Now choose two variables from the dataset and describe one, what potential variation might be when you might find in those two variables. Two, if there is any measurement error and which one is it. Three, if there are any adjustments that you think you might need to consider before analyzing the data. Please look at nutritional variables instead of demographic variables. Let's wrap up this presentation and summarize the main key points that we just learned. So first, remember that variation in diet intake follows consistent patterns, and we need to account for each of them. Remember that there are different variation types, such as random, systematic, within person, and between person variation, and that each one of these can affect the associations that we're trying to study. So, for example, if we're looking at random variation, then our results might be attenuated. So it means that we might not see any result. With a systematic variation, we can have bias in any direction. So that means that our results might be higher than they should be or lower than they should be. And then that within person and between person variation can also affect the results, as we need to have multiple people in our sample, but also we need to have multiple measurements for each individual. Let's also remember that measurement error must be considered and handled appropriately during the analysis stage. But before that, we need to make sure that our assessment tools are appropriate to reduce this measurement error, and that even before we decide what tool we're going to use, we need to know what is our research question and variables. This will prevent to have biased results. Also remember that there are multiple sources of measurement error, not just variability, and that some of these can be misclassification on the individual's usual intake, and these could be due to human error. And then there can be reporting errors, such as under and overestimating the nutrient and food intakes. Let's also remember that energy intake is determined by body size, physical activity, and metabolic needs, which can also be related to disease risk, and therefore energy intake must be adjusted when we analyze nutritional data. But it's going to be case by case depending on the research question, as sometimes we just need the absolute value intake instead of this relationship. And finally, something that we have discussed thoroughly. Remember that there is no ideal assessment tool, and sometimes they might be inadequate to examine some of the associations. So again, before we decide upon which one we're going to use, let's make sure that it's reproducible, that it's valid, and that it actually accounts and collects the data that we need to study the associations that we want. Thank you for your time, and if you have any questions, just let us know.